Structure, digestion, and transport of dietary lipids. Plan of the lecture: the biological functions and classification of lipids, digestion of triacylglycerols, absorption of dietary lipids, synthesis of chylomicrons, transport of dietary lipids. Biological functions and classification of lipids. According to the structure and functions, we can divide lipids into several groups. The first one is storage lipids or neutral lipids. They are triacylglycerols, which contain glycerol and free one type or different type of fatty acids. The second type is membrane lipids or polar lipids, phospholipids and glycolipids. They have a difference in structure and contains of alcohol or disaccharides. Glycerophospholipids contain glycerol to fatty acids, phosphate group, and alcohol. Sphingolipids contain sphingazine, alcohol, sphingazine, one molecule, one residue of fatty acids, phosphate group, and choline. Glycolipids could be divided into two groups, sphingolipids and galactolipids. Sphingolipids can also contain sphingazine, fatty acid, and mono or oligosaccharide. Galactolipids contain glycerol, two residues of fatty acids, mono or disaccharide, and sulfate group. Lipids are hydrophobic molecules, and one of the most spread of lipid structures is fatty acid. Fatty acids consist freely of esterified to glycerol, which contains 16 or 20 carbon atoms in length and may be saturated or unsaturated. Saturated have no any double bonds. They have only one bonds between two different carbon atoms. Unsaturated lipids or fatty acids contain double bonds. When these residues of fatty acids interact with lipids, we can produce monoacylglycerols when glycerol contains only one residue of fatty acid, diacylglycerols, which contain two residues of fatty acids, and triacylglycerols, which contain three residues of fatty acids. In structure of phosphoglycerides, one residue, one OH group, and the structure of glycerol removed interacts with phosphate group and choline, which gives a specific semi-hydrophobic and semi-hydrophilic properties to build membranes. Different membranes like cellular membrane or intracellular membrane, like complex Golgi complex, membranes in mitochondria, and different others. Sphingolipids contain specific alcohol sphingazine, which form hydrophobic tail in the structure of these types of lipids. Another different type of lipids is cholesterol. Its specific structure contains four rings and hydrophobic tail, which supports structure of membranes. Another type of lipids are eicosanoids. They work as signal molecules and produced by action of oxygen, a specific enzyme, cyclooxygenase, onto unsaturated molecules in the structure of membrane. They work as signal molecules. Cholesterol also works as a structural compounds, but at the same time it can convert it into specific signal molecules, steroids. Digestion and absorption of dietary lipids. The main idea of digestion of lipids is cleaving bonds between different residues in the small intestine. This reaction catalyzed by a specific enzyme which produces pancreatic gland and called as lipase. Lipos from Greek means fat. And after production of small components like glycerol, monoacylglycerol, or free fatty acids, they transported into the epithelial cells of small intestine. After that, they form triglycerols, which have specific phospholipid envelope and the specific protein which inserted in this envelope and transported in the bloodstream and transferred by blood from small intestine into other organs. For digestion, in organisms of animals and humans, takes place a production of a specific molecules, bile salts. They work as emulgators, which they dissolve big drops of fats into small drops, which help us to increase rate of digestion by lipase. After using of these bile salts, we reabsorb them into our bloodstream and reuse them in our liver and convert them, and after that, after secretion by gallbladder into the small intestine. For transport of lipids, we use specific molecules. We call them micelles. They have an envelope, phospholipid envelope, supported by cholesterol molecules. Inside these structures, we can find fatty acids, free fatty acids, not much. Not a large amount, and triacylglycerols. Envelope has also specific proteins which can give information our cells about contents of these structures. To produce micelles, we use specific transport system and synthetic system. We use Golgi complex to interact of proteins and phospholipids, which produce in different sites. And proteins for synthesis of micelles we produce in roof and the plasma reticulum, and phospholipids for envelope, for transport form of lipids, we produce in smooth and the plasma reticulum. Golgi complex works as a site of production of the whole structure of micelles, and after secretion, this micelle transported into the blood, or previous site in small intestine into lymph, and after that into blood for spreading of these lipids, for transport of these lipids by blood from small intestine to liver, muscles, and different other organs. Blood lipoproteins. We have several types of lipoproteins. The first one, which produced in small intestine, is the chylomicrons. They produce in small intestine and transport delivery and transport the dietary lipids from small intestine to other organs. Chylomicrons, micron remnants, are converted into the small structures and transported into the liver. Very low density lipoproteins produced by liver and they transport endogenous lipids. After conversion into intermediate density lipoproteins, low density lipoproteins, they transport cholesterol or some components or precursors of cholesterol to organs which use cholesterol for synthesis, specific components of membrane-free cholesterol, or they use cholesterol for synthesis of different hormones. High-dense lipoproteins, we have several fractions of this transport form. They provide reverse transport of cholesterol into the liver from peripheral organs. Chylomicrons are synthesized in intestinal epithelial cells, so their triacylglycerols are delivered from dietary lipid, and their major apoprotein is ApoB48. Chylomicrons travel through the lymph into the blood, and different other proteins like ApoC2, the activator of lipoprotein lipase, and ApoE are transferred to nascent chylomicrons from high-density lipoproteins, and mature chylomicrons are formed. In peripheral tissues, particularly adipose tissue, muscles, the triacylglycerols are digested by lipoprotein lipase and transported into these organs, so as a form of free fatty acids. The chylomicron remnants interact with the receptors on the liver cells and are taken up by endocytosis and reused to produce another components for transport form of lipids.